And I would like to introduce our second speaker, Professor George Sugihara. George is currently a McGowan Chair Professor of Natural History in the Institution of Oceanography, Scripps Institution of Oceanography, excuse me. George has worked on many disciplines. He's a mathematical biologist. He worked on food web problem, climate research, fisheries, neuroscience, cardiologist, just to name a few. George has many titles. One unique title that most ecologists do not have, but probably that he has been managing director for Dutch Bank. So through his work with ecologists and banker, he actually suggested that banker should also learn ecology. Today he will talk about the prediction, coupling, and causation. Welcome, George Sugihara.
Thanks for the kind introduction, Zach, if it were only true. Okay, so I've actually added one word to the published title, nonlinearity, and I actually think that's a pretty important one. So let's just get into it. Hold on a second here. I just click. Okay. So I think one of the important problems of nonlinear systems, we all pay lip service to the idea that nature is nonlinear, but we tend to study it as if that's not true. And I think what happened in the economic crisis is one example of that. A lot of our risk metrics were based on observations that were made during what's called the golden decade, sort of the 10 years preceding the crisis. And during that time, the amount of variability that we saw in things like GDP, earnings growth, was pretty small. And if you form ideas of risk based on that small variability, you'd say something like 5 sigma should never happen. Okay? But now, one of the characteristics of nonlinear systems is that they tend to be heteroscedastic. So the variances tend to be very irregular. And if you, this is data that was given to me by the Bank of England. If you go back 150 years, you find that what we thought was 5 standard deviations is really about 1 standard deviation. So the large changes that we saw that we thought should never happen actually do happen quite a lot. There must be a better way to click across. Push which one? F? Oh, okay, got it. All right, so technologically deprived here. I'm missing the gene. All right, so here are 2 time series. These represent maybe 2 species. And I think it's a reasonable hypothesis to say that that these things are correlated or coupled. Now, if you look at this, if you observe it for another, say, 15 years, you find they start out being coupled, but then there's a little bit of disruption there in years 48 through 55, 51, and then all of a sudden it snaps back into place. So, I mean, if we're ecologists, we'd say, well, that's a pretty good hypothesis. We were right, most of the time, that these things are positively coupled, they're correlated, they're causally related. But if you look at this over really long periods of time, and again, I haven't changed anything, this is essentially the same system, you would find that your hypothesis falls apart. You have periods of where they seem anti-correlated, periods where there's no correlation at all. But in fact, these are from a couple, 2 species difference equation. So I haven't changed anything. All I'm doing is I'm looking at different periods of time. So again, one of the real, what seems like a hassle or a problem with these nonlinear systems is that they have this tendency to produce what I call mirage correlations. So you tend to get to see a pattern that makes you think you understand what's going on, but then all of a sudden the system seems to flip into another regime and that pattern disappears. And flipping into another regime doesn't necessarily mean that anything has changed. It just means it's a nonlinear system. That's just how they behave. Okay, I'm going to give you another quick example, sort of a more graphic one. This is the famous Lorentz system for modeling convection rolls in the atmosphere. So just a toy model. Just think of it as three coupled differential equations, where if you vary one parameter, it affects the other. Okay, just like an ecosystem. Okay, so there are the three time series. Now, if we try the normal thing that we do in science is look for correlations. Okay, and if we look for correlations here, we find we don't find anything that neat. This is actually with 10% of observational noise added. But if you're looking at a correlation between x and y or x and z, and especially between y and z, you find nothing at all. Okay, but even worse. So this is over very long time periods. Even worse. This is again that same Lorentz attractor or the butterfly attractor. If you look at motion in one of these wings, is there a pointer? Yeah. Yeah. If you look at motion here, you'd say that these two variables, y1 and y3, are negatively correlated. Okay. Now, if all of a sudden the system flips into this wing, again, nothing has changed. All of a sudden, they're positively correlated. So it's just the nature of nonlinear systems that you're going to get this correlation is going to fool you. But correlation is what we do in science. And there's a real disconnect here between saying nature's nonlinear and then using correlation to try to understand nature. Okay. Okay. This is a problem that goes back to Fisher-Barkley. Said correlation does not necessarily imply causation. And then as a corollary, we just saw lack of correlation also doesn't imply lack of causation. So with those two coupled species, they appeared to be uncorrelated, but in fact, they were causally connected. So not a lot has happened in 300 years. Granger causality was the major new innovation, and he said prediction. He suggested prediction as an operational criteria for causation. And this actually won him the Nobel Prize in economics. Okay, let me, hold on. I have to go back. How do I go back? Which one? R. Okay. So this is a little animation. Actually, the animation is a chance to give me a 4-minute rest that explains, I think, some of the essential ideas behind an alternative way to look at this problem. So it's one thing to criticize. It's another thing to offer something constructive. And I think this is possibly a constructive alternative.
This is the Lorentz attractor. Lorentz is an example of a coupled dynamic system consisting of 3 differential equations, where each component depends on the state and the dynamics of the other 2 components. So you can think of each component, for example, as being species, foxes, rabbits, grasses, and each one depends on the state of the other one. So these y sub I that we are calling the axes are actually the state variables, or the Cartesian coordinates that form the so-called state space. The manifold M consists of the set of all trajectories, and phi is the flow on M defined by the coupled equations. X of t is a point on the manifold. So what's interesting here is to view a time series then as a projection from that manifold onto a coordinate axis of the state space. So here we see the projection on axis y1 and the resulting time series recording displacements in y1. This can be repeated on the other coordinate axes to generate other simultaneous time series. So again, these time series are really just projections of the dynamics occurring on the manifold into coordinate axes. So conversely, we can recreate the manifold by projecting these individual time series simultaneously back into the state space to create the flow. In this panel, we can see the 3 time series, y1, y2, y3, each of which are really a projection of the motion on that manifold. And what we're doing is the opposite here. We're taking the time series and projecting back into the original 3D state space and recreating that structure, which is the manifold, the butterfly attractor. There's a very powerful theorem that was proven in 1981 by Flores-Takens that shows generically that one can reconstruct a shadow version of that original manifold simply by looking at one of those time series. For example, consider the 3 time series shown here. These are all copies of each other. They're all copies of variable y1. Each is displaced by an amount tau from the other. So the top one is unlagged. The second one is lagged by tau. The blue one on the bottom is lagged by 2 tau. So Taken's theorem says that I should be able to use these 3 time series as my new coordinates and reconstruct a shadow version of the original butterfly manifold. So let's see how this works. So, this is the reconstructed manifold produced from lags of a single variable. And you can see that it actually does look fairly similar to the butterfly attractor. It's the same in terms of topological invariance, in terms of important mathematical properties. And it represents what's known as a one-to-one mapping between the original manifold, the original butterfly, and this reconstructed version, M prime. This is, I think, the most important aspect of these reconstructions, that in fact, generically, there is this one-to-one mapping between the reconstructed manifold and the original native underlying manifold, which is the butterfly attractor in this case. We use this fact to show how causation between time series variables can be established by investigating whether or not they belong to the same dynamic system. This is accomplished by testing the relationship to a common underlying manifold. This practical criterion for detecting causation is based on reconstructing manifolds for suspected causally related variables to see if indeed they belong to the same underlying manifold and are therefore functionally related.
Okay. So these are, I think we ended up actually with the wrong one here, but so these are examples of taking this particular set of time series and projecting them in 3 dimensions. A bit confused here. So just to reiterate. Okay, I guess this is, I ended up with a different PowerPoint, but this one's all right. The real simple idea is that you can take a time series, and then from this time series you can create one of these kind of shadow reconstructions that has essentially all the same topological properties as what the original dynamical system had. And the whole idea behind looking for causation between variables is to show that they belong to the same dynamic system. And if that's the case, then you can show that from one variable, I can make one of these shadow manifolds and use that to tell me something about the state of the other variable. And so that's basically how this idea works. We're not looking at correlation, but we're looking at some dynamic, there's some sharing of dynamic information between time series. Okay? So just to show you that you can actually do this with real data. These are salmon time series provided by the Canadian Department of Fisheries and Oceans. And they're desperate to figure out how to forecast salmon. Turns out this method works pretty well. So if you do one of these really simple lagged reconstructions where you take this data and you take a fork with 3 prongs and you lay it down on the data and you call one prong X, the other one is Y, the other one Z, and then you plot that point and then you move the thing forward and you plot that point and plot that point and you keep doing that. This is what we get. And so, in fact, we end up with something that looks kind of like an interesting coherent geometry out of a time series that looked like a bunch of junk. And that's a fairly common thing that you can see with these nonlinear time series. That in fact you get these time series that look like noise, like utter junk. But if you do one of these embeddings, you actually get some interesting geometry out of it that suggests that there is an underlying manifold. Okay. So now, in fact, natural systems are not these I don't think that they're low dimensional. I think that they're really high dimensional, but I also think that you can approximate them reasonably well in relatively few dimensions. So, for example, those salmon attractors may in fact be 100-dimensional, but we can approximate them and get some reasonable predictability in that case in just 3 dimensions. So this is the idea that this is not an exact description of nature, but it's an approximate description, and it's one that you can verify by using forecasts, and in particular out-of-sample forecasts. So here's prediction. We have a particular way of predicting using sort of similar past histories in order to predict the future. I mean, this is how we all make predictions. But we have a particular protocol that involves dividing the time series in half, ideally, and using one half to make the manifold, and then the other half to predict. So we basically make the model with one half, and then we test it by predicting points that it hasn't seen. So if we do that on this time series, so this looks like an utter mess. So we divide this in half, and now we embed this in 3 dimensions. So it's just taking a fork with 3 prongs, laying it down, and dragging through and seeing what we get. This is what we get. Okay, it's something that's fairly coherent. Now, if we use this thing to say, I'm here now, all these points in the past, then one over there, to make that kind of a prediction, this is actually what we get. We do extraordinarily well. This is actually predicting two time steps ahead. So from something that looked like white noise that from a statistical point of view had no information in it, we actually can make reasonable predictions. It's kind of like magic, but it actually does work. All right. So how do you know that we should have used a 3-pronged fork? Well, the way you do that, and not a 2-pronged fork or a 6-pronged fork. So the way you do that is by looking at how well you can predict. So this says that if I have 3 dimensions, my predictability rho is the correlation coefficient between observed and predicted. If I use 3 dimensions, I actually do reasonably well. And it starts to fall off as you add more dimensions, because what you're doing is adding irrelevant information. So you get confusion in the nearest neighbors that get chosen. Okay. And there's a relationship between the dimension of the object that you're creating and the dimension that's needed to hold that object. It's called the embedding dimension. I won't go into that. All right. So here's a real-world example. This involved data that I actually got from a colleague in Australia, Maria Milicic, who's actually a New Zealander, but she was at Lizard Island. And she read our paper and saw that it had these time series that looked like garbage. She said, that looks just like mine. And so she sent her time series to us, and we looked at it and found indeed it had some kind of a signal. So here are the time series that she sent. This is larval supply. These are palmocentrid larvae caught in light traps at Lizard Island. And they have this kind of strange spiky look to them. And if we do one of these embeddings, we actually find that we get a row close to 0.8 with 3 dimensions. So that was pretty encouraging. I mean, from essentially not being able to explain anything about those ups and downs to all of a sudden getting and this is again doing this out of sample, getting a row of 0.8 was pretty good. So what this tells you then is that it's a low-dimensional or at least can be approximated as a low-dimensional nonlinear process. And it says that you can do this with 3 variables, which is kind of encouraging because it says that we have a chance of actually making some kind of a model if we could figure out what those 3 important variables are. So the idea then is to actually try to make a model based on that idea. So we constructed what we're calling a mechanistic embedding using basically what we had is a bunch of parallel time series. And we said, well, if we do that trick of trying to combine these parallel time series until we get one that seems to make sense, that produces some kind of coherent geometric object, then maybe that's a way of identifying the real variables. So we tried that. It was kind of a trial and error sort of exhaustive search. And we actually found something, but the thing that we found I thought was garbage. I thought, well, we just tried everything and we got something that seemed to fit, but that doesn't mean that we had a model that really worked. And in particular, we found this thing actually made sense because these guys spawn on the full moon, and so this nighttime illumination lag 19 days made a lot of sense. Now, and this variable also made sense. Cross-shelf wind speed lagged 1 day. It's best if we don't lag this at all. Basically, it says that the larvae are being blown into the light trap versus being blown away. But the thing that really bothered me was that these moderate wind speeds needed to be lagged around 16 days. And that one didn't seem to make any sense. And so I told Maria that this is, I think we just curve fit here. And she got all excited and said, no. These things are born with a yolk sac, and it takes 3 days to absorb the yolk sac. So on day 16 here, on the lag day 16, on the third day, they are most sensitive to, their yolk sac is absorbed, and they're most sensitive to current conditions and good feeding conditions. And so she said, moderate wind speeds is sort of in theory and in the literature what people are saying is important. So it felt good to kind of reverse engineer and actually come out with, by doing a trial and error search, getting something that actually made sense biologically. All right. So, and this model actually ended up explaining a lot of the variation. So, okay. And that's what the attractor looks like if you do one of these lagged reconstructions. Okay. I want to just quickly, I want to try to speed up here because I think we're running out of time. This is a 5-species model. The point that I wanted to make here is that you look at these things and sort of there are times where they seem correlated and there are times when they're not. If you look long enough, none of them are really very correlated. So you can see that these are pairs of species and there's no real correlation here. Again, this is a nonlinearly coupled system. But it turns out that you can use one species, you make a manifold for one species, and you can use it to predict the other. So that's pretty convincing evidence that these things are actually dynamically coupled. Okay. Now, this is doing the same thing, but using all 5 species to make the embedding. And you can see that actually works better. All right. So, and it turns out that these multidimensional ways of constructing models gives you a lot more power, and it also allows you, in this case, to identify which groups of species are interacting and which aren't. We call these functionally coupled units. So I'm going to skip here now, in the last 3 minutes, to talk about causality. And Granger suggested using prediction, remember, to address what he recognized as the insufficiency of correlation. Now, and so this is actually what I'm going to show you is something from a paper that's in press. I'm sorry, it's currently in review in Science. Wishful thinking. I hope it's in press. It presents a new paradigm that's different from Granger's. Granger's doesn't actually work very well for ecological systems. And if you look at the actual definition of Granger causality, he's saying that suppose you have a universe of all possible variables, and I'm trying to predict y2, and the variance of y2 is pretty small, so I'm doing very well at predicting y2. If now what I do is I remove some variable y1 from the mix, then because the model got worse by taking y1 out, I can say that y1 Granger causes y2. Okay? So, again, Granger's assuming that I can remove a causative variable, and if the predictability goes down after removing that causative variable, that causative variable, that's how you define a causative variable by Granger. Okay. So the problem is the Takens theorem, which is what we have been using, and Granger's idea are incompatible. So Granger's idea insists that you can actually remove a variable. that this condition, but this condition is not feasible in a dynamic system, okay? Because all the information is contained in the target variable itself, in the lives of the target variable itself. So there's a fundamental problem. Granger assumes that this is possible, but this is not possible in a dynamic system. Okay? It's possible in a stochastic system, but not in a dynamic system. So if you believe that ecosystems, for example, are dynamic systems, then Granger causality doesn't work. So we need something that works for dynamic systems. And we make a new definition, basically, dynamic causation. We say that if two variables they're causally related if they belong to the same dynamic system. So it's like in the Lorentz attractor, Y1 and Y2 were related because they belong to the same dynamic system. And in fact, we could use Y1 to predict Y2. So that's this basic idea of cross-mapping. We can use if from one variable we can get information about another variable, dynamic information about another variable, then that's a way of showing that they're causally related. Okay. So, and the particular method that we've come up with is called convergent cross-mapping. And basically what it does is it uses it uses the history of y is the affected variable, to recover past states of the causal variable. Okay? Really different from Granger. Granger focuses on causes. What we're doing is focusing on effects. We're saying, if a variable is affected, then it ought to contain information about the things that are affecting it. And that information, that imprint, should be extractable using this funny method that I've been talking about, basically constructing these manifolds and then using them to predict. So let's see how this actually works. One of the things that happens here, if this is true, so if x is affecting y, then we should be able to use this y time series to construct a shadow manifold to recover the states of x. Okay? And a necessary condition that this is actually happening is that this estimate of x, this recovery of x, should get better as the time series length increases. So that's one of the necessary conditions here, and what we call this convergence. So here you can see rho, how well the observed and predicted estimate of x is, given that I'm using y to predict it, with time series length going into y, going in to construct that manifold for y that's now being used to predict the past values of x. So you can see the predictability goes up. This is convergence. So let's see how this actually works on data. I think that's the most interesting one. All right. This is the absolute most classic experiment in ecology. It's the old Gauss predator-prey experiment, right? Where we're using didinium as the predator and the prey is paramecium. Okay. Okay, so the blue is paramecium and the red is didinium. All right. Now, what I'm going to try to do is take this didinium time series here and try to extract information about paramecium from it. See if I can use the didinium to give me information about paramecium. So that's what this is. That's what happens here in the blue, and you can see that there's some information encoded in the didinium time series that actually allows me to recover the past states of paramecium. Okay? I can do the same thing now for paramecium, using paramecium time series to cross-map didinium. I'm using the paramecium time series and I'm asking, how much information is there here that's really being reflected by the dynamics going on down here? Okay? This is not just correlation. And I'm finding that, in fact, because didinium affects paramecium very strongly, there's a really strong signature in here about what didinium is doing, which means that didinium is having a very strong effect on paramecium, which basically means top-down control, so which is also consistent with this experimental protocol. So we actually can reconstruct the degree of causal strength, coupling strength here, by using this convergent cross-map technique. So you can see that, in fact, this shows top-down control by the predator on the prey. Okay, here's another example. This one is not quite as clear-cut, but it's a little bit more interesting. There was a sort of a historical debate that spawned the California cooperative fisheries investigations in 1949, and what happened, of course, was the collapse of the anchovy fishery and the downfall of Cannery Row. It turns out that Cannery Row was built during a period when which was actually kind of an anomaly in terms of the abundance of sardine. But there were initial hypotheses that if you look at over the 20th century and you look at worldwide, there tends to be some anti-correlation between sardines and anchovies. So that suggested that they were competing, and there were a number of people who made that formal hypothesis. On the other hand, other people have said, no, they're not competing, but they're actually being forced by some common outside entity. So again, this is this idea of a Moran effect. How can we detect a Moran? How can we distinguish whether two things are actually interacting or whether they're being forced by something from the outside? So luckily we had the data and a kind of a technique that allowed us to get some insight in here. So what we found is, so the obvious thing is to see, first of all, if I make a manifold for sardines, can I learn anything about anchovies? The answer is no. Okay. If I do the same thing for anchovies, can I learn anything about sardines? Again, the answer is no. So there's no information about each species, about any dynamic interaction between species, any causal interaction actually contained in their time series. So even though they're negatively correlated, kind of. Okay. Now, on the other hand, if you look at sea surface temperature, and now what you're doing is you're using sardine time series to try to reconstruct historical sea surface temperature. Turns out you can do it. It's very bizarre. Okay, so this shows that in fact there's information embedded in the sea surface in the sardine time series about sea surface temperature. Okay, so I'm basically using sardines to forecast or to recover what the past states of sea surface temperature were, and that works. You can again, you do it for sardines. Again, you see there is some information, but it's not that strong. It's there, but it's not all that strong. So, I mean, this is kind of interesting because it says the way these things are regulated right now is that they have it's actually written into state law that there is a fixed temperature that's associated with fixed pier, with Scripps pier, that if the temperatures are above or below 17 degrees, the harvest targets change. So this says it's not a matter of a fixed temperature, but what we really need to understand is the relationship of temperature to abundance. Okay, so the final thing I'll do real fast, since Zach already pointed at his watch, is complexity. And I just want to say that this kind of view that I've been trying to introduce to you, and that there'll be a number of presentations during the conference that will elaborate on this and hopefully make it clearer, is sort of antithetical to the way that we normally like to analyze systems. Normally we like to, and this is true in banking as it is in fisheries. In fisheries, people do single species management. They pay lip service to ecosystems, but in fact, they manage one species at a time, as if you could, reduce the universe that way. And banks do the same thing. They say, I'm not interested in how the financial ecosystem looks. I'm interested in my firm's risk. So it's called single firm risk management. Exact same mindset, and it's just the way that we as human beings prefer to think, even though it's wrong. At least we can do it. It's wrong, but we can do it. These techniques actually allow you to embrace complexity as opposed to try to simplify it away. So I put this at the end because I wanted to provoke people. I figured this is going to make at least half the people in the audience mad. I think the reductionist paradigm is something that we all tend toward, but it also has given rise to the single species view, and it's given rise to the view that, sure, we should have ecosystem-based management, but that's going to be too complex. I mean, we're going to end up with things like EcoPath and EcoSim and all this stuff, and we're never going to be able to parameterize all this stuff. And the other thing is that this idea that even though we say that it's nonlinear, we say, well, suppose that there's an equilibrium, we can approximate it as linear in some neighborhood. And if you can do that, then you can decompose it into parts. Then Granger's idea would work. Okay. And the other thing is that stationary statistical phenomenology should work, but obviously we know it's not going to work in a nonlinear system. So I think perhaps the emperor hasn't. So we would really wish that the world was stable and decomposable, but in fact I don't think it is. Right. So this also demonstrates I think that I have no taste.
Thanks for the excellent talk. We have time maybe for only one quick question. If there's a... Yeah, please come forward to the microphone. I cannot really see anyone.
Thank you very much for that very exciting talk. I just had a question about the length of time series required to do this sort of analysis, and also in the testing of the model. Because if you've got a number of different variables, whether they're species or temperature or these other things you're talking about, but on top of those there's longer term cycles, decadal, annual, longer term climate cycles, just for an example. Do you have some sort of indication you can give of the time series you need to reconstruct the manifold? And when you do have that time series, when you split it into the two sets, do you split first half, second half, or do you need to subsample randomly from the original time series?
That's a great question, actually. So, in fact, ideally, you would want to just split it in half. And the early stuff that we did with epidemiological time series are all split in half. But we're finding the time series we're dealing with now are really too short to do that. So we do tend to subsample, and or we do cross-validation even, where we remove the point that we're trying to predict and neighboring points that may be included in any vector that would be used in the library. So that's a great question. And ultimately, the answer is, how much data do you need? As much as possible, right? But here's the other part of it. Because if indeed you have a complex system where you have these interconnected parts, you can actually leverage the amount of information present in any one time series. So for example, if you only have 50 data points in one time series, or let's say you only have 30 data points in one time series, if you have, say, four different variables that you've been measuring simultaneously that you think may be interacting, then the amount of information grows factorially with that. So instead of having on the order of, say, 30 or 40 vectors, you could end up with 70,000 vectors. So the amount of information that you get, because nature is complex and interconnected, is huge.
Thank you.
Thanks.
Thank you very much. I'm sorry we are wrong out of time. George will be still around for the rest of the week. So if you have questions, please ask him during the sections. Okay, so we are closed our plenary section. And thank you very much. Thank you for all the speakers. Enjoy your lunch.